All right, we'll go ahead and get started. Good morning or good afternoon, depending on where you're at. Good evening if you're joining from outside of the US. Thank you guys, everyone, for joining us for our first MPRINT webinar of the fall. This fall, we're going to do something a little bit different. We're going to have 2 webinars a month. The first webinar on the first Tuesday of the month will be at 1 p.m. We'll be focused on fellows and early career investigators, trainees, and give you some basics of skills that you will need to know to succeed in clinical pharmacology career in maternal and pediatric therapeutics. So today I'm going to start looking by talking about some components of non-compartmental population pharmacokinetics and physiologically based pharmacokinetics, just kind of giving everyone an overview of the various techniques we can use to evaluate PK data and support PK analyses, and understanding when it's appropriate to use what analysis method. So this is meant to be a very interactive webinar. We actually have some discussion questions for the last part of the webinar, and so feel free to raise your hand if you have a question or enter into the chat box, and I'll try my best to address it as time comes up. So that will get started. So the objectives of today's talk are to describe a variety of pharmacokinetic modeling techniques and to understand the advantages and disadvantages of each technique. I've listed here the various techniques we will be discussing. These include non-compartmental analysis, empirical compartmental models, such as naive pool, 2-stage analysis, and population PK approaches, and then physiologically based pharmacokinetic modeling. And we'll also touch upon allometric scaling and its use for pediatric pharmacokinetics. Finally, we'll determine and discuss through some case series the appropriate pharmacometric approaches that can be used to evaluate various pharmacokinetic data and how you would design a PK study for a variety of scenarios. So first, for those new to the area, I'm sure we have a wide audience here today. Why are we talking about pharmacokinetics and why is it important? Pharmacokinetics discusses quantifies the movement of drug throughout the body. We usually think about this quantification in the plasma, but it's also important to think about the movement into tissues, particularly target tissues. And as you watch the video here, you can see the injection of drug into this mouse and how the drug will move apart as the highlighted in the brightness into the kidney and the heart and the brain. So it's important to know to use pharmacokinetics and enable personalized or precision medicine, and to determine the accurate dose needed to help to treat a condition, to get above a minimum effective concentration, but to stay within the therapeutic window and prevent toxicity. So you can see in the curves on the right here shows 3 pharmacokinetic curves demonstrating scenarios where in the blue curve A, you see that there's a very high peak concentration that gets above this therapeutic window, maximum tolerated concentration, and may lead to extra adverse events. In case C, there's a more extended release of drug, and we see much lower concentrations so that we're not able to obtain a minimum effective concentration. However, in case B, you can see that the majority of the time, for about 8 hours, the drug is above that minimum effective concentration, but it does not go above our maximum tolerated concentration, and so it's a much safer dose for the patient. So pharmacokinetics through this helps you determine how much or what dose is required and how often it should be administered. to maintain concentrations within that therapeutic window. Drug disposition can be quantified throughout the body. So in the dose of the drug is always remaining, is the total amount of drug administered to the body. And we can identify where this is because it will fall into various compartments. The dose can be pushed into the amount of drug at the absorption site, plus the amount in the body, the amount of drug excreted, that metabolized, and the metabolite excreted. So you'll see as we initially give drug, it moves from an absorption site into the body where it's metabolized, and then a component of the metabolites and the parent drug may be excreted or eliminated. And so the law of mass balance says that the total amount of drug in these various sites among the body will always be equivalent to the dose. So what we're going to talk about today is you've developed a PK study in your population, and you have a set of data. For instance, here you see individual concentration time curves, profiles for a number of individuals across 6 hours following administration of a drug. So we're going to talk a little bit about what do you do with it now? How do you actually analyze the data, and what methods make sense, how you would look at the data. In general, there's two methods that we'll talk about. One is non-compartmental modeling or NCA analysis, and the second is compartmental models. Non-compartmental models are based upon the statistical moment theory, and really you can just plot your data and use general algebraic equations to solve for the AUC, observed Cmax and Tmax, and calculate an elimination rate constants. This incorporates a standard set of formulas for determining PK parameters such as clearance and volume. Compartmental models, on the other hand, fit the data or fit a model to the given data. So you'll have your observed concentration time profile, and you can select a 1 or 2 compartment method to fit that data, and then the model defines the PK parameters. So going a little more into detail on these approaches, in the non-compartmental approach, you will plot your data and connect to the curve. And so this is just showing a general concept of a concentration time profile. When you plot this data on a semi-log plot, so you have log concentration versus time, you can take the slope of the line to estimate the elimination rate constant, or the KE. And you can use what we call the trapezoidal rule, which is demonstrated here, to estimate the area under the concentration time curve profile. You can also just visually observe Cmax and Tmax from your data. And then using this information and the dose, you can calculate the clearance as dose over AUC, or dose times bioavailability over AUC, and a half-life, which is the natural log of 2 over your elimination rate constant, as well as the volume of distribution, if you know what that elimination rate constant is. On the other hand, when you are using an empirical pharmacokinetic model or a compartmental model, your body is represented as a series of empirical compartments. So in this case, we see an absorption phase going into the central compartment and the clearance phase. So this would be a compartment with uniform distribution, fairly quick within that compartment. And we use first order rate constants generally to describe distribution and elimination from these models. So what you do here is you take the data and you fit the parameters for your model to that data. So you have your observed data and you're pulling down, or you're fitting a curve to the data. So you can look at various compartmental models with various compartments. For instance, a 2-compartment model would have a quick distribution phase, such as shown here in 1, and a slow distribution phase 2 in this component, so a central and peripheral compartment, which would then take into account distribution into a plasma and then a slower distribution into the tissue compartments. And these can be, these compartmental models can be written either as an algebraic equation, as seen here, or as a set of differential equations as seen on the right. So no matter what type of model you're using, whether it's a non-compartmental or compartmental model, there's some various ways you can use you can fit the data to it. One method is to use naive pooling. In this way, you have data points that from a variety of individuals. You can see here on the right, each of the data point, each individual is represented by a different symbol and color. But when you're fitting your model, you're assuming that all the data points arise from a single individual. So you fit your curve to all of these data as a single function. Well, this gives you a mean profile. It ignores inter-individual variability and correlation in time within individuals. A similar method to this would be to fit the average profile for each individual. So instead of taking all your data, putting it into a program and fitting a single compartmental model to that data, here we will take the average of each of those data points first. So for instance, you're taking the average at Cmax, average at 0.5, at 1 hour, 2 hours, and so forth, averaging each of those into a single point, and then estimating your parameters from this mean profile. One of the issues with this is that measurements must be made at the same time points across all of the individuals, or you may see some variation in your curves across those individuals. Historically, the standard 2-stage approach was used for fitting PK parameters to compartmental models. In this approach, each of the individual is treated as treated separately. You fit a model to those individuals, and then you take the sample mean and covariance from that total data. So you would calculate a clearance and volume for each of these individual curves, and then take the mean, the averages of those parameters to get your population estimates. Well, each of these has value. You can see that when you're comparing the methods used, you do get different results. So in these examples I've shown previously, we've simulated data with the true values shown here of a clearance of about 20, volume of 108, and Ka of 0.84. Using the standard 2-stage approach, we recover nearly those identical parameters. However, when we're using naive pool and average fit methodologies, you see that the clearance is underestimated and the volume is overestimated, as well as the Ka. So you see there are some variability when you're using these approaches that you need to be aware of. There are also a number of limitations for all of these methods. First, they all require densely sampled or rich concentration time data to adequately capture the pharmacokinetics in the individuals. As noted for the naive pooled, you need to first calculate the mean concentration, and then for each of the individuals, you need to have it. It also does not deal with missing data. So for instance, consider estimating a PK profile where we're missing this Cmax concentration in an individual, and how that would make a difference when you're calculating your AUC profile for an individual with or without that Cmax component. And you would see that there's you'll get very different estimates for your PK. And it may also be difficult to deal with variable dosing regimens and nonlinear pharmacokinetics and other sources of variability in these data. So in order to capture that population variability, we can turn to using a population pharmacokinetic modeling or PopPK approach. This approach uses a statistical method of nonlinear mixed effects modeling to simultaneously fit data from multiple individuals to estimate between and within subject variability. So some of the advantages of population PK over the two-stage and prior approaches we've discussed is that it does allow for sparsely sampled and missing data, and that samples from the same individual are correlated. And this allows us to better estimate those within and between subject variability aspects. You can also account for variability in dosing and nonlinear PK using these methods, as it does take into account dose in a different aspect. Oops. So overall, when you're looking at developing a population pharmacokinetic model, you're looking at first identifying the typical values for PK or PD parameters of the drug and response within a population by characterizing a population mean. So you can see in the plot on the top, we're just capturing the population mean values for this curve, the population curve. You can also explore systematic differences in PK by employing covariates. In this case, you can see that there's two separate groups here, one in the gold color and one in the red color. The red color having a lower clearance than the group in the gold color. So looking at potentially a drug interaction or a pharmacogenomic effect leading to differences in clearance here. And finally, you can still estimate an individual PK parameter by fitting information on intra and inter-individual variability to the model and capturing individual components and PK parameters through that method. And I'm not going to go into detail on how you would fit a population PK model, but I do think it's important that you understand the components behind a PopPK or a nonlinear mixed effect model. And so we'll spend a few minutes discussing that. First, each PK model will have a structural model. This will identify the fixed effects, or theta is another term that's used for those. These are simply your PK parameters, your clearance, your volume, your Ka, and they're determined as a function of the concentration versus time curve as a function of those parameters, the dose, and the time. This can be these parameters can be determined using either algebraic or differential equations. On top of those, on top of this structural model, then with the fixed effects, we then add random effects or a stochastic model that captures between subject or between occasion variability. And we term these omega, or the eta is the variance for the omegas. And you can see the difference. The etas would be if you have two different subjects here, the difference between their PK profiles and the mean profile is captured within these eta terms. Another source of random effect is residual unexplained variability, or your sigma and eta terms, and these are, or epsilon terms, and these are the unexplained variability that may be due to differences or issues with an analytical assay or other aspects of the PK parameters that aren't captured by your covariates and your intersubject variability. And finally, as I mentioned, you can talk about covariate effects, this can be added, covariates can be added on your thetas as well as on your random effects terms. And so you can see that even within the individual groups of male versus female, there's also variability within those that can be captured. This slide summarizes what you will see in what we've talked about in the previous slides, that the population pharmacokinetic model will describe your plasma concentration profile as a function of your fixed effect or your theta terms, your clearance, volume, and other covariates, with these residual variability and between subject variabilities, the random effect terms, as a function of dose and time, and can incorporate other covariates. So you can see through these, we're able to then describe and capture a pharmacokinetic model for not just the population, but also for each individual patient. This slide shows the difference between the traditional empiric models that we talked about previously and population pharmacokinetic modeling. Traditional pharmacokinetics works best in healthy volunteers or highly selected patients that have very similar PK profiles or expected PK profiles, whereas population PK can really work better for a more targeted patient population. So we will often see these in our pediatric patients. It's also used quite often in elderly and organ dysfunction patients. Traditional pharmacokinetics, again, requires very dense sampling and it's typically conducted in small size studies with partly due to the cost of that dense sampling. And there's minimal inter-individual variability expected between the individuals. Whereas in population pharmacokinetics, these are typically larger studies with fewer samples per patient. Now, part of this is because the fewer samples you have per patient, the more data, the more number of subjects you're going to need to actually fit your population pharmacokinetic profile. There can be a wide variety of inter-individual variability in demographics, pathologic, physiologic conditions, co-administered medications that can cause drug interactions. And you can also, population pharmacokinetics tends to be a little bit more commonly used for predicting future events and evaluating relationships between PK and PD. But there's often times when we don't have sufficient clinical data to fit a population pharmacokinetic model or a non-compartmental or empirical compartmental model. And often these studies in pregnancy and pediatrics can rely on physiologically based pharmacokinetic modeling. PBPK modeling, unlike PopPK modeling, is a bottom-up approach. So we're using PBPK to predict concentrations based on physiochemical and in vitro data and prior data that we know about a drug, versus in population pharmacokinetics or empirical models, we have data that's observed and we're fitting a model to that data. So in PBPK, we have a structural model that looks more like a typical body structure, right? So we have various organs where each of the organs represents an actual physiologic compartment. And then we combine compound specific parameters such as molecular weight, log P, pKa, in vitro determined clearance, distribution aspects, protein binding, drug interactions, and receptor binding, with the physiologic or system parameters such as tissue weight, tissue blood flow, enzyme abundance, transporters, and tissue composition, and so forth, to predict what will happen to the drug in the body and to predict the PK. One advantage of this is that it allows you then to integrate a variety of data on your compound as well as your system to get a better mechanistic understanding of the pharmacokinetics. So there can be a variety of PBPK model structures. Again, typically they represent the compartments in the PBPK model represent various organs and tissues, but there can be a variation in this, and you can see some models will be minimal or lumped PBPK models that do not necessarily reflect physiologic organs. You could still have your central and peripheral compartment volumes that come from, say, data from a clinical study. Unlike the empirical PK models, which tend to be more of a mammillary model, these organ compartments are connected via the venous and arterial blood flows, as you can see here, with the arterial flow going into the tissue and the venous flow coming out of the tissue. What your model is structured as can be determined by a variety of information you have available, such as the goal of the model, what core tissues, fluids, and organs you are most interested in. If there's a tissue of special interest, excuse me, for instance, in pregnancy, are we specifically interested in fetal concentrations or placental concentrations? You'll want to know if there's a tissue that accounts for a significant amount of the drug, and another consideration may be tissues with available experimental data. One of the keys to PBPK modeling is using in vitro and vivo extrapolation to assess the clearance of a drug. In these, this will allow us to take data from an in vitro metabolism assay in S9 fractions or microsomal fractions, scale that information, the intrinsic clearance gain from the in vitro assay, to a intrinsic clearance per gram of liver using scaling factors and corrections for enzyme abundance, and then further scaling it on into the liver intrinsic clearance by multiplying this by the microsomal protein per gram of liver. And finally, based on the liver weight, we can estimate a total hepatic clearance. Typically, this is done using the well-stirred model that is shown here in the purple box at the bottom. PBPK is commonly used in drug development across all spectrums, from preclinical through late development and into stage 4. In the preclinical realm, we often are using PBPK and estimates from in silico predictions of clearance and volume to determine drug disposition in preclinical species. This can then be scaled to help support our first in dose in healthy volunteer studies and early development. And then in late phase, we can add in some of those system parameters and look at information on drug interactions, pharmacogenomics, age, organ dysfunction, pediatric and pregnancy populations to get more of an idea of what the pharmacokinetics of the drug will be in these special populations. PBPK is recognized by regulatory agencies and is shown here is commonly used in drug labeling and development. This shows 3 different data sets. The first up to 2014, the bottom here up to 2016, and then on the right, a data set that was collected from 2018 and 2019. And you can see that while the majority of PBPK is still being utilized for drug-drug interactions with enzymes or transporter, a significant portion is also being used to look at pediatric drug development or other areas as well. Again, as I mentioned previously, PBPK tends to be a bottom-up approach where we take information on the available data from physiochemical properties, in vitro assays, and potentially some in vivo preclinical ADME or early clinical phase PK to project disposition of the drug in human, allow for information on first in human dosing, enzyme and transporter drug interactions, and then in general provide a mechanistic understanding that supports use of these models in special populations. This compares to the top-down approach, which utilizes the clinical concentration time profiles and to estimate the PK parameters. Here, top-down approaches, the PopPK approaches can really be useful in helping identify inter-subject variability and covariates to support clinical decision-making. But these can also be combined kind of in what we call a middle-out approach. So a middle-out approach utilizes both the clinical data and the PBPK model structure. So we start by having our bottom-up PBPK model and then refine it based on our clinical data in order to capture things like renal function and the effect of renal clearance on a drug, or potentially looking at special populations such as pediatric or organ impairment. So with that, I will conclude kind of the lecture portion and we'll move to the interactive portion of our webinar today. First, I will ask if there are any general questions on what I've discussed so far. If not, we will move into some of our case studies. And in these case studies, what we will go through is how, some of the areas of drug development where you might see these various PK studies designs utilized. So as we're moving through those, think about how we might use the different types of analyses that we've discussed. So non-compartmental analyses, empirical, naive pool, or two-stage analyses for compartmental models, what would fit a population pharmacokinetic analysis, as well as PBPK. We'll also talk a little bit in the next section about the use of allometric scaling. So we're going to start with development of a new drug. So a company is developing a first-in-class drug to treat ADHD. You know the structure, the in vitro clearance, and the in vitro protein binding. But they want to know, what dose should I give to a mouse in order to obtain a therapeutic effect? So how would you initially start, only knowing structure, in vitro, and in vivo data in modeling this data? What approach could we use here? And feel free to turn on your camera and speak up.
Most probably we can go for PBPK modeling.
All right, yeah, so this would, knowing only the in vitro information, we would have to use a PBPK type modeling approach where we're putting together our drug data and the physiologic system of a mouse.
This is
Is there anyone like to share how you would design a study like this? What would I want to know as I'm dosing my mouse for the first time with this drug? So my first question might be, how do they want to dose the drug? Is this going to be something that's administered intravenously, orally, or subcutaneously? We'd want to know then what formulation would be used. And then we'd also be interested in trying to figure out when we would draw some blood samples. So there's some variety of options you can use to look at sampling times and how this would happen. And some of the work I'm involved in, we will draw multiple samples per mouse. And so we can create an individual PK profile in a single mouse. In other studies, we will use a mouse for a single blood draw, maybe sacrifice the animal and get tissue concentrations at each time point. And so then we'll have to use an approach that would allow us to take the mean data from each of the animals. So the naive pooled approach or mean data approach to evaluate the PK for these animals. So I've kind of already talked about this. What I would want to do is to look at the PK. Typically, we would start with a non-compartmental approach, assuming that we have sufficient concentration time curve profiles to evaluate the PK of the drug in an animal. And this would either be used using a naive pooled approach, a mean data approach, or if possible, an individual PK approach. Typically we will hear from investigators that my mice are all the same, so we don't need to worry about the variability in them, but we have found cases where there's quite a bit of variability in the same strain of mice treated at the same age, which is also something to keep in mind. So we've moved on. Now we may be closer to what many of you actually deal with in your real world lives, and first in human dosing. So we've completed a series of PK/PD studies in animals, and the company is ready to move to phase 1 clinical trials. How would we determine what dose to administer based on our animal data? I will give you a hint. So a lot of places will use allometric scaling.
Yeah.
And so this simply is a method of moving from species to species, or within humans, from an adult to a pediatric patient based on body weight. And the clearance of a drug is in volume are correlated with the body weight. And this is based on the heat production of the metabolic rate in a variety of animals, from very small single cell organisms up to large elephants and whales, and finding that they're all in a log linear relationship. So this is one way that we can use our empirical models or our non-compartmental analysis to scale from an animal to a human. Another way to identify that first in human dose would also be to use physiologically based pharmacokinetic modeling. We can look at the differences in vitro clearance between our animal and our human microsomes, and then changing our system parameters, estimate what the PK would be for a human based on our animal data. And both are commonly used in industry now to move forward. So we'll move just a little bit closer. Now we've got our drug studied in humans, and we find that it's a CYP2D6 substrate. So how would you design a study and using PK modeling to help determine the effect of CYP2D6 inhibitors?
Well, to come back to what you were saying before, when you're first working on humans, would you not want to start off conservatively at a lower dose and then build up to find out when you're going to exceed your dose thresholds?
Yeah, I kind of skipped over those. I went from first in human to now we know what dose we're using, but correct. You would usually in your first in human dose, you would start at a conservatively low dose to be able to obtain your data, because, you know, while we're fairly confident in some of our modeling techniques, we're not 100% with them. And, you know, a lot of times our errors, you know, within twofold may not be sufficient for a first in human dose. So we want to make sure we're staying within safety guidelines.
Also, another thing that I didn't understand from your original most studies is, why wouldn't you go immediately to the 2-compartmental model? I mean, what is the advantage of the earlier models?
For the PBPK models?
Yeah.
Yeah, so you can take data from the in vitro and use in silico methods to predict what the distribution will be through those PBPK models. So you can use like a Poulin and Thiel method for determining volume of distribution, or and take data from your microsomal studies to help determine clearance. And so that will give you a nice prediction of what your animal data will look like. And this is before you have PK data, anything observed in that animal. Now, once I get the animal data back, I'll look at it using typically a non-compartmental approach first. And if needed, then maybe a compartmental approach.
I have a question regarding the previous slide. No, not the allometric scaling, the PBPK. So when you go from animal to human, so are you trying to achieve similar exposure in the human being too? So is that your goal? You're trying to achieve this, achieve similar exposure in human being given the physical chemical properties and the difference in physiology? Is that what you're trying to do? Correct.
Yeah. So if you have a pharmacodynamic endpoint you've identified in mice, and you know the constant, or and that you know what your concentration at the site of action should be, then you can use the PBPK modeling approach and changing your system parameters to match a population of humans. You can then estimate what concentration or what dose you would need to supply those concentrations.
Okay.
Great. Are there any other questions?
Yes, this is from my side, Yasin. Just I would like to go back when you're discussing about the different methods of like new pooling and two-stage methods. So there you are showing like a true method or true values. So what exactly mean by true value?
So that the true value is what was used. Those were simulated data sets, and the true value is what they used to start those simulations.
Okay.
So that was what we expected.
Okay, so means how do we calculate, using the PopPK method, or what exactly method we are using to calculate that values?
Those were what, so if it was simulated based on a 1-compartment model, that was the mean and standard deviation that was used to determine to do a Monte Carlo simulation of the other of those 6 individual data sets and that.
Okay, thank you.
Yep. Great. So what are some options when you're designing a study to look at drug interactions?
Shall we do with or without inhibitors and see the exposure response?
Right. So typically you're looking for a study that is designed with or without the inhibitor and with your substrate drug, so the drug that we're developing here. Are these typically small size studies or really large studies?
Initially, probably we can go for a small size studies.
All right, so initially we're looking at doing a relatively small clinical trial, typically 12 to 20 patients, where they're given the drug with and without the inhibitor or inducer, the interacting drug, for a period of time. And we typically collect relatively extensive sampling from these individuals. So if we have extensive sampling, what would be some options for how we would analyze the data from these studies?
One way would be to probably use the PopPK. If you got the genotype available, maybe you can find the clearance and check the effect of the genotype on the clearance.
Yeah, so you could use PopPK, but in general, we have a fairly limited sample size, and extensive sampling. So typically these studies would be analyzed using more of a non-compartmental or an empirical compartmental approach, assuming that dosing is equivalent. If you have a larger sample size, then and looking at something like genetics, which I think is my next question of how would you look at the impact of pharmacogenomics, which I think is what you were discussing more, is in this case, then a larger sample may be necessary, where we're able to get sufficient genotypes from each individual. And then in that case, you may want to have a larger number of subjects. And so you may not be able to afford, from a cost standpoint and it's ease of carrying out the study, it may be difficult to get or not feasible to get a large number of samples across a dose range, but you could get sufficient number of samples to do a population PK study here and look at the pharmacogenomic effect as a covariate on the clearance. Great. All right, now I just have 2 more slides that get into kind of general discussion of things we would want to look at when we're talking about pediatrics. So what are some options for study design and analysis of PK data in pediatric patients? So we have a lot of data now on our adult patients. How would we initially look at this, look at what the pharmacokinetics are in pediatrics?
PBPK modeling approach might be useful.
Yeah, so right, a PBPK modeling approach. What are some considerations here? We talked about allometry, which would be useful as well if we don't have a full PBPK model. But the PBPK approach would allow us to change not just the size of the patient in general, but we can also look at changes in development of enzymes and transporters that can also affect the pharmacokinetics in those younger patients as well. So a lot of times they'll utilize PBPK modeling. Great. So if you're looking at a set of really young patients, outside of, obviously I'm a PBPK person, so I like using PBPK, but now we want to look at actual concentrations in these children. Are you going to be able typically to study in a neonate, say, to get a lot of blood volume from these individuals or a number of samples?
We certainly are not, and we would have to take a scavenged sample approach that would utilize other blood samples that were drawn for clinical care. and perhaps combine those in a population-based approach to gather more data.
Exactly. So we would be looking more from, we'd be less likely to use our non-compartmental or empirical compartmental individual approaches and need to use more of a population approach, because it would take more patients with fewer samples to assess our PK. So great. And I will also say this is where PBPK modeling can help, because you can help provide the aspect of mechanistically does it work, and if you're able to take those samples and confirm your PBPK model, it's helpful. Okay. Does anyone have a specific question about study design for pediatric patients? Not. We'll move into the next set, which is pregnancy and lactation studies. So pregnancy and lactation studies historically have most of the data, at least in pregnancy, is a single time point during pregnancy. Some studies have gotten data from the pregnant individual at various time points, maybe 3 or 4 times throughout pregnancy, and then postpartum. But one of the things that we also have to think about with pregnancy is that it's not just a static change in PK, that there are physiologic and changes in drug metabolism that occur across pregnancy and change throughout pregnancy and in the postpartum. And so it's really important here to take into account the timing of getting the samples, not just following drug administration, but also timing during the pregnancy itself. One thing we'll see with a lot of pregnancy studies is that we'll use opportunistic sampling, where a woman would be taking a drug chronically, and then you can get PK samples at multiple time points during the pregnancy because she's already on the medicine. Many of these have more dense sampling, and so they have been analyzed using methods like non-compartmental analysis. A few studies have done population pharmacokinetics, but they're relatively limited in scope. Another thing you will see with studies in pregnancy that you need to be careful with is in the studies at delivery, a lot of times you'll see something like a cord to maternal plasma ratio reported. Those are also dependent on dose time of the drug, and so it's also important to take that into account, because the PK in the plasma may have a different shape than the PK in the cord blood, and so you will, or an offset. And so depending on the time after dosing, you can get a difference in concentration. So, and the same is true with lactation. The time when you sample your breast milk and your plasma sample may lead to differences in that milk to plasma ratio, depending on the pharmacokinetics of the drug. So these are really areas where PBPK modeling can be helpful in order to look at more mechanistically the changes in PK across pregnancy and during lactation. Anyone have any thoughts or discussion questions on that? Great.
Does PBPK analysis is also used for formulation type of analysis?
For formulation?
Yep.
Yeah, so you can use PBPK to help support formulation studies, and there's methods to extrapolate from in vitro breakdown of the formulation to the clinical PK of it as well. And you can look at a variety of formulations, not just oral. I know Dr. Bees has done quite a bit of work on vaginal delivery of dosing forms as well.
Thank you.
With that, I will close our webinar today and thank you for your attention. I will also note that the MPRINT Pharmacometrics and Clinical Trial Design Core is available to help support trainees or others who have questions about optimal trial design for pharmacokinetic and pharmacodynamic studies in maternal and pediatric populations. And feel free to reach out through the website or by email at info@mprint.org or directly to me as quinney@iu.edu. And we'll be glad to answer any questions you have and set up discussion with you. And I will also invite you to attend our next MPRINT Hub webinar. This will be a more scientific nature webinar on Thursday, September 15th at 2 p.m. Eastern time. So thank you for your attention, and we look forward to seeing you next time.
Thank you.